Let's put a little summary onto cellular respiration and oxidative phosphorylation. In aerobic cellular respiration and oxidative phosphorylation, the final molecule at the bottom of the electron transport chain is oxygen. Microbes that use this particular pathway are called aerobic. There are also anaerobic microbes that do cellular respiration and oxidative phosphorylation. Not just strict, obligate anaerobes, but other oxygen requiring microbes as well. Remember, there are also facultative anaerobes and there are aerotolerants. So anytime a microbe is doing this anaerobically, they're just using a different molecule at the end of the electron transport chain, something that's not oxygen, something like nitrate or sulfate or carbonate. Something else to note here. Microbes that do anaerobic cellular respiration and oxidative phosphorylation are going to get a little bit less ATP from their sugar than they would if they were doing it aerobically. In other words, that's where the variation comes from when I say somewhere between 30 and 36 ATP get made for each simple sugar. The variation comes from whether or not this is being done with oxygen as the final electron acceptor or with one of these other molecules, the nitrate or the sulfate or so on. Oxygen at the end of the electron transport chain is the best. It's just the best, and you're going to get the most ATP out of that. That's the 36. If you're doing it anaerobically with a different molecule, you're going to get a little bit less ATP, somewhere between 30 and 36. It's still a very efficient mechanism, whether you do it aerobically or anaerobically. It's very efficient to do oxidative phosphorylation. It's also very important for us to continually remind ourselves that microbes that are doing anaerobic cellular respiration are not fermenting. Fermentation is a completely different mechanism. Anaerobic cellular respiration and fermentation are not the same thing. During cellular respiration and oxidative phosphorylation, whether it's aerobic or anaerobic, little bits of ATP are going to get made along the way using substrate level phosphorylation. Remember, that's during the glycolysis portion and the Krebs cycle portion. Large amounts of ATP are going to be made during the oxidative phosphorylation portion. This is extremely efficient. This is a good way to make ATP. You're going to get between 30 and 36 ATP from your simple sugar when you use oxidative phosphorylation, and you'll get a little bit more from the substrate level phosphorylation. All cells require ATP and use ATP for energetic coupling. Now let's spend a couple minutes talking about fermentation. Remember, we said in cellular respiration, you have to have a final electron acceptor molecule available at the end of the electron transport chain. And that's because there are electrons coming down that electron transport chain, and electrons have to have a place to go. They were released from somewhere during oxidation, and now they have to be received by a molecule in reduction. So if you are a microbial cell that is doing cellular respiration, you've got to have your final electron acceptor available to you in order to keep doing that kind of metabolism. If you don't, the electron transport chain is going to stop. It's not going to keep going. because oxidation and reduction must occur together. So the electron transport chain is just going to stop transporting electrons. And that means no hydrogen ion gradient is going to get built using the energy from those electrons. And that means no ATP is going to get made using oxidative phosphorylation. That is a life-threatening problem for the cell. If it is faced with a situation in any moment where it doesn't have its final electron acceptor available, it has to stop using cellular respiration to make ATP, and that's life-threatening. Cells will then convert over to a secondary pathway, to their next choice for producing ATP, for their next choice metabolically speaking. And that's when a cell will begin using fermentation. We talked about in the laboratory slides, we talked about how fermentation allows the first part of respiration to keep going. It allows the glycolysis part to keep going. And during that glycolysis portion, a little bit of ATP is getting made through substrate level phosphorylation. So when a cell stops doing cellular respiration in that emergency situation, it'll keep making pyruvate. It can, it's able to. It's just that the pyruvate can't keep heading down the same pathway. The pyruvate is going to need to be redirected along a secondary metabolic pathway called fermentation. So you and I, for example, our cells are going to do cellular respiration at any time they're able to do cellular respiration. But if we find ourselves in a situation where we either don't have oxygen or we don't have enough oxygen, our cells are going to keep making pyruvate, but they're going to send the pyruvate down a fermentation pathway temporarily. We said, if you have a heart attack and you fall down to the ground and you stop breathing, that's a life-threatening event. And the reason it's life-threatening is because you're not taking in any oxygen in that moment. So respiration cannot continue for you in that moment. And you have only a few minutes worth of ATP stored up. So fermentation can kick in temporarily for you, but it's not going to save you because it's a secondary pathway. It's not an ideal pathway. It's not going to make enough ATP for you. We also see fermentation in humans not when there's no oxygen, like the heart attack example, but when there's not enough oxygen. For example, the person who is running a sprint, or the person who is exercising to a degree they're not capable of. And you've seen this person, and you may even be this person once or twice in your life, where you get into an amount of activity where you're basically gasping for breath because you're working so hard in that moment. You're not getting enough oxygen in that moment. You're not able to do respiration efficiently in that moment, so you will start to ferment. And remember, the two most common fermentation pathways are the lactic acid pathway and the ethanol pathway. And humans do the lactic acid one when they ferment. So your body will temporarily do a little fermentation, make some lactic acid, and lactic acid is quite irritating to our muscles. But it will keep you alive in that time where you're not getting enough air, you're not getting enough oxygen. It's different in microbes. Yes, there are some microbes that are just like us, but there are other microbes that, well, they may switch back and forth pretty readily between cellular respiration and fermentation. And then there's a third group of microbes that just use fermentation. So again, they've got a lot more options than we do, depending on the species that you're talking about. Lots of microbes use fermentation relatively frequently, and some of them use only fermentation. Fermentation is producing ATP for the microbe. The microbe is doing it to get the ATP. But along the way, they're producing some kind of an end product. And remember, we said those are alcohols or acids or gases. And there's a variety of them. There's a variety of different types of alcohol that are made depending on the species of microbe. And there's a variety of different acids that get made. And there's a couple of different gases that get made depending on the type of fermentation pathway and the species of microbe that you're talking about. Lactic acid fermentation and ethanol fermentation are just the most common pathways that we see. We already looked at these pathways in lab, but let's just review them here. In lactic acid fermentation, the simple sugar is going to go through the first part of respiration. It's going to go through the glycolysis part to produce pyruvate. The pyruvate molecule is then going to be used to make lactate. So there's a particular enzyme that will do that for the cell. Take the pyruvate and chemically make lactate from it. And I just want to point out here, if you're not familiar, when we talk about acids, we name acids with this ATE at the end. So yes, you can call a molecule lactic acid, or you can call it lactate. So pyruvate is actually an acid. You could call it pyruvic acid, or you could call it pyruvate. Those are some nomenclature things to know about acids. So in lactic acid fermentation, because glycolysis is still going on, that's how we get from the sugar to the pyruvate. That means substrate level phosphorylation is going on, and we're getting some ATP as well. Alcohol fermentation is a little bit more complicated. There's just an extra step in alcohol fermentation. The sugar will be converted to pyruvate. Then the pyruvate will be converted first to a molecule called acetaldehyde, and a little bit of CO2 will be made in that reaction. And then the acetaldehyde will be converted to ethanol. So there's an extra step in alcohol fermentation, but it's just another fermentation pathway. And because the sugar is becoming pyruvate, substrate level phosphorylation is going on, and a little bit of ATP is being made along the way. The last two slides in this lecture are just diagrams of lactic acid fermentation and alcohol fermentation. Let's walk through the lactic acid diagram together. This represents the glucose molecule, or it could be any other simple sugar that a microbe is going to ferment. But let's use glucose right now. Glucose is going to be oxidized in this process called glycolysis. Remember, glycolysis isn't one reaction, it's actually 10 reactions. And at the end of glycolysis, we're going to have pyruvate made. Glucose has 6 carbons in it, so at the end of glycolysis, we're going to get 2 pyruvates because each pyruvate has 3 carbons in it. So 2 3-carbon molecules come from a 1 6-carbon molecule. Along the way of glycolysis, substrate level phosphorylation goes on. And ADP will have phosphate groups added to it to make ATP. And yes, it says 2. We're going to get 2 ATP made here. Don't worry about the numbers. Just know that substrate level phosphorylation is going on as glycolysis goes on. So now we have pyruvate, and we can't send the pyruvate into pyruvate processing and Krebs cycle because we've gone into an alternate pathway now. So instead, the pyruvate is going to be used to build lactic acid or lactate. Now there's other details in this drawing. They're showing us that the electron carrier, NAD and NADH, are being involved here. Don't worry about that detail. Just know that the pyruvate will be converted into lactic acid in this particular form of fermentation. And finally, we have a diagram representing alcohol fermentation. Here's the glucose again. The glucose is being broken down into pyruvate. A little bit of ATP gets made along the way. The pyruvate then is processed into acetaldehyde and a little bit of CO2. And the acetaldehyde will then be converted into ethanol.